Hello everyone, Zia on this side. A brief introduction of mine. I've done MSc Zoology, have qualified CSI and JRF with all India rank 65. Have qualified SET Maharashtra as well, and one more exam that is called as PET for the PhD. Currently I am doing my PhD at SPPU, Pune. Now, our today's topic is DNA replication. What is DNA replication basically? So it is a phenomena in which a duplicate copy of DNA is synthesized. It is a phenomena in which a duplicate copy of DNA is synthesized. So here the two strands of the DNA helix separate and each strand acts as a template for synthesizing a new strand. So basically, like if I'll be drawing, I'll be just going for this, like here I am having like this kind of DNA, and again like this strand, another strand is forming, and on this strand another strand is forming. So this is the case of the DNA replication. And after the completion of the replication, the two copies are produced which will be having one parental and one newly synthesized strand. Both of them will be having one parental and one newly synthesized strand. So this is a scheme of the replication and also called as semi-conservative mode of replication. Semi-conservative mode of replication, because one of the strand is parental, which is a conserved one, and the other one is a newly synthesized, which was like just formed. Now, the experiment which was to prove that the DNA replication is semi-conservative, it was proposed by scientists called as Michelson and Stahl. It was proposed by the scientists called as Michelson and Stahl. So they have used one of the most important microorganism of all time, that was none other than E. coli. Normally, like the nitrogen is N14. Normal nitrogen is N14. But they used a medium which was containing heavy isotopes of N15. They used a medium which was containing heavy isotopes of N15 as a nitrogen source. So basically, ultimately, they have to take that N15 as a newly synthesized DNA strand. So ultimately, their DNA became heavy DNA. So this heavy DNA was differentiated from the normal DNA by density gradient centrifugation, because a normal DNA will be having N14 and this heavy isotope DNA will be having N15. So there's an experiment called as DNA gradient centrifugation using the CaCl, or what we call as cesium chloride, as the gradient. So this was the case. Then again, like this heavy nitrogen containing E. coli were again transferred into the medium which was having 14 nitrogen as a nitrogen source. Now this sample was taken from the media and again the DNA was extracted. Now what happened here? What is the division timing of the E. coli? It is 20 minutes. E. coli divides every 20 minutes. So basically here now, like after 20 minutes, we were having the DNA which was a kind of hybrid mixture. Basically, this was something like one of the strand was having N15, and the other strand which is newly formed will be having N14, So this was the case after 20 minutes. When they continued the experiment after even 40 minutes. What happened there? That the DNA extracted after 40 minutes had equal amount of hybrid and light intensity. And like they had continued that experiment again and again, and now every new strand which was forming was having N14, only N14, because only N14 was the available nitrogen source. So ultimately each and every strand was having N14. So somehow they proved the case that the newly synthesized DNA is obtained only on the one strand only, and the other strand is the parental. So that was the case. Again, like I'm showing you the same DNA molecule. This parental was N15, whereas the newly synthesized was N14. So this is like hence proved, they should say, like the replication of DNA is semi-conservative. Hopefully this replication semi-conservative phenomena you have understood. I'll be dealing with the proper replication phenomena in the next lecture. Thank you very much.